So hello and welcome to Only Sai No File. Don't forget to like, comment, share and subscribe. In this video I'll be talking about protein misfolding and prions. Now we very well know that a protein to execute its function correctly should be properly folded into its three-dimensional structure. And if this does not happen, that is, if proteins do not undergo proper folding, that is, undergo misfolding, usually lose their function. So, what cells do is either refold them into its correct structure or denature and degrade them. Now, what happens sometimes is this misfolded structures form prions. Now, what prions are? These are infectious agents which usually aggregate and form insoluble complexes which escape the mechanisms that help refold and degrade the proteins. Now, basically, what happens is prions are usually transmissible. I'll be talking about how they are transmissible in some time. And these prions usually cause diseases such as mad cows, scrapie, CJD. So normally you may have heard about bacteria, viruses, fungi causing infections or diseases. Now, this is an example of proteins, that is misfolded proteins, prions, causing diseases or infections. Now, suppose this is a normal protein, PRP, in the brain whose function is related to the brain synapses or the neuronal synapses. So, what happens is this is a normal PRP, call it C protein. PRPC is a normal protein which has alpha helices. Now, what happens is due to some kind of mutations or some changes, it is folded or misfolded, you can say, into another form which is PRPSC. Now, SC refers to scrapie. So, this normal protein PRPC is misfolded because of some changes or mutations in the misfolded PRPSC proteins. Now, the basic difference between this is this usually consists more of alpha helices, whereas the misfolded protein consists of beta sheets. Significance of this beta sheet is that they have a potential to aggregate. Now, what usually this PRPSC does is, if because of some mutation there is a formation of PRPSC proteins, that is, suppose there are three PRPSC proteins, these with via non-covalent interactions form aggregates. Now, what basically happens is, as I mentioned, usually the brain has these PRP protein. What happens is there is a pool of PRPSC proteins as well as PRPC, that is normal proteins, in the brain. By mechanisms still unknown, this PRPSC proteins react with the normal PRPC proteins, and usually they convert this PRPC into PRPSC proteins. That is, they usually spread and cause misfolding of another normal proteins also. So these eventually, after this PRPSC proteins are formed, what they usually do is they even form larger aggregates, that is, they form amyloids. They are called as amyloids, which are insoluble in nature. And now what they do is, since they are present in the brain cells, that is neurons, they cause infection and destroy the nerve cells. That is, what these proteins do is they are not refolded back. They escape mechanisms that are involved in refolding or even degradation, and therefore eventually via a series of steps causes destruction of nerve cells and cause this infection. Next is, now this overall destruction of the neurons or the nerve cells and infections degenerates the brain function. And even more surprising or shocking is that these prions or misfolded proteins even transmit to other individuals. As I already mentioned about mad cow and scrapie, what generally happens if there is a certain animal which is already infected with this particular prion protein, and if an individual consumes that particular meat, there is a high chance, or rather, you can say, that individual is at a risk of getting that particular disease itself, as these prion proteins are transmissible in nature, and therefore they transmit from one individual to another. So, this was all about prions and misfolding.